This video will cover the photosynthesis light reactions. Photosynthesis is generally divided into two parts. There are the light reactions, which require light as a source of energy, and then there are the dark reactions. And although the dark reactions are driven by the products of the light reactions, the dark reactions don't technically need that light energy as an input anymore. So we'll just be looking at the light reactions portion of photosynthesis. But first, a little bit of background about photosynthesis as a whole. Most autotrophs do photosynthesis, but autotrophs fall into two different categories. There are what we call chemoautotrophs, which derive energy from inorganic molecules. And then there are photoautotrophs, who derive energy from the light coming from the sun. So autotrophs doesn't necessarily mean photoautotrophs, although it is generally what we mean, especially if we're talking about any kind of a plant or alga or protist, and even some photosynthetic prokaryotes like cyanobacteria fall into this category, the photoautotrophs. Of course, though, photosynthesis is such an important process because photoautotrophs tend to make up the base of most food pyramids. And so although photoautotrophs are really the only ones who do photosynthesis, just about every living thing on Earth depends upon that process. So here we see the general chemical reaction that describes photosynthesis. Remember that photosynthesis, like cellular respiration, is not really just one chemical reaction. It's actually a series of many chemical reactions, but this is what we would call a summary reaction. So the inputs for photosynthesis, the reactants, are carbon dioxide and water. And in this diagram, you can see how the reactants become the products. So in the products here, we see glucose. That carbon in glucose comes from carbon dioxide. So does the oxygen in glucose also come from carbon dioxide. A lot of people assume that because they're both gases, the oxygen that's produced in photosynthesis comes from the carbon dioxide that goes in. That's not actually the case. The oxygen that's produced in photosynthesis actually comes from the water that goes in. We note light energy here. It's not technically a reactant, but it is a necessary input to drive the reactions of photosynthesis. You notice that this is an endergonic reaction. We're taking smaller molecules, simpler molecules, carbon dioxide and water. We're combining them to make glucose, which is a much more complex, energy-rich molecule. So the light energy going in is an essential part of the endergonic reaction. We could also describe this as being anabolic because a larger molecule is being made. It is also non-spontaneous. So where do those reactants come from? Well, carbon dioxide, as you probably know, is taken from the atmosphere. Plants can take up carbon dioxide from the air and then water from their roots. This slide summarizes some of the key players in photosynthesis. Some of these we'll take a look at in a bit and in more detail. We have the chloroplast. This is the organelle where photosynthesis takes place. All of photosynthesis takes place within the chloroplast. Light, of course, coming from the sun. We'll take a closer look at what it means to be visible light in just a moment. There are certain pigments. These are molecules that are responsible for capturing light energy. And we'll see that pigments are really what make plants so special, what makes them able to carry out photosynthesis where other organisms cannot. The electron carriers in photosynthesis are very similar to the electron carriers we saw in cellular respiration, except that NAD+ is now NADP+. It's called that because it has an extra phosphate group. You can remember, though, that you've got like a P there, sort of because you've got photosynthesis happening. It's a trick for remembering it. It's not actually true, but you can remember that the P in NADP+ is only present in photosynthesis. Just like in cell respiration, when NADP+ picks up 2 electrons, it is reduced to NADPH. Finally, we have photosystems 1 and 2. These are complexes of many molecules that include some electron acceptors, like we saw in the electron transport chain of cell respiration. Also chlorophyll, the pigment, the primary photosynthetic pigment that traps light energy, and of course some proteins. We'll look more closely at these photosystems in just a moment. So let's jump back and take a closer look at the chloroplast. Remember, this is the organelle where all of photosynthesis takes place. You should be familiar with the general structure of a chloroplast. Notice that inside the chloroplast, which has a double membrane, we have an additional membrane system called grana. So these grana are like little discs, except that they're not quite discs because they're not discrete. They're actually continuous. So you can sort of follow the inside of the grana like it's a maze. The grana are made up of individual layers. Each layer is called a thylakoid, and the space within the thylakoid is called the thylakoid space. The space that's inside the double chloroplast membrane, but not quite inside the grana, this is called the stroma. And the structure of a chloroplast is very important because we're going to learn that different parts of photosynthesis occur in different places. And so you'll need to be familiar with the architecture of the chloroplast. So where are chloroplasts exactly? They're pretty much any place where a plant is green, but they tend to be most plentiful in leaves. So you don't find too many of them in stems, for example. Leaves are essentially designed to be little solar panels, right? They're flat and they absorb a lot of light energy from the sun. And so that's where you tend to find the chloroplasts. Now, if you were to look more closely at the structure of a leaf, if you were to look at a cross-section of a leaf, what you would see are a lot of chloroplasts along the top of the leaf. We call that the palisade layer. And then more loosely packed chloroplasts along the bottom of the leaf. And that's simply because at the top of the leaf you have a lot more light energy coming in, so you want to cram that with as many chloroplasts as possible so that you can make the most of that light energy. You'd also see a few other structures on the leaf. So here you have stomata. These are little pores, usually on the underside of the leaf, that allow oxygen to come out, carbon dioxide to go in. They also allow for the release of water through the leaves. Here in this diagram, you can also see a vein running through the leaf. Veins you can see from the outside of a leaf. These will be the xylem and the phloem running through to deliver nutrients and water to the leaf. Let's take a closer look at the light that strikes the chloroplasts. As you know, the electromagnetic spectrum includes a lot of forms of light, most of which we cannot see. But the visible light that hits around this wavelength here, 10 to the third nanometers, includes all of the different colors of light that we can see. So we've got short wavelength light, which means higher energy here in the violet area, all the way to red light over here. Red light has a longer wavelength and thus it carries a lesser amount of energy than the violet light does. Any of this light here will be usable by the plant. And it's usable by the plant because plants, or photosynthesizers in general, contain pigments. So these are special chemicals that can absorb certain wavelengths of light. At the same time, though, they will transmit or reflect other wavelengths. So if you consider the shirt that you're wearing right now, let's say that it's like a navy blue, for example. The reason that it looks navy blue is because your sweater is absorbing all of the different colors of visible light that strike it from the sun. The sun's light is white, which means it contains all of those visible colors. So your sweater or your shirt, your navy blue shirt, absorbs all of those colors except for navy blue. Navy blue is the light that is reflected off of your sweater. It's the light that was reflected off of your shirt that hits the eye of the viewer, and so the viewer perceives your shirt as being navy blue. This is what a pigment does. It absorbs some wavelengths of light, but reflects others. Chlorophyll is the primary photosynthetic pigment in photosynthesizers in plants, and it reflects green, not too surprisingly. So in this diagram here, you can see white light coming in from the sun, bouncing off of the chlorophyll pigments, which are located in the thylakoids, and all of the colors of light are being absorbed except for that green light, which is being reflected. The consequence of this is that plants can use all the colors of light for photosynthesis except for that green light. The green light gets reflected, and so it's not actually captured by the plant. It is reflected and unusable. Let's take a closer look at those pigments. I'm going to jump down here to the light reactions. So chlorophyll A is the main photosynthetic pigment, but there are what we call accessory pigments that help in photosynthesis. These would include chlorophyll B and the carotenoids. The purpose of the accessory pigments is to make sure that that green light that's reflected by chlorophyll is not completely lost. Chlorophyll B and the carotenoids tend to be different colors from chlorophyll A, and so they can absorb a little bit of that green light, not all of it, but they can absorb a little bit of that green light so that plants aren't totally lost, or so that that light energy is not totally wasted. Plants can use these accessory pigments, but they're not going to replace the importance of chlorophyll A, the main photosynthetic pigment. So if you were to grow a plant under a green light, it would be very unlikely to thrive, because a green light would be depriving the plant of almost all of the pigments, or all of the light color they can actually absorb and use for photosynthesis. So green light, not so good for a plant. Here you can see the structure of chlorophyll. It has a hydrocarbon tail. Remember, this structure here means lots of carbon-hydrogen bonds. But the real action center is what we would call the porphyrin ring. So this is the head of the molecule. And you'll notice that at its center you have a magnesium atom. And the difference between chlorophyll A and chlorophyll B is really just a very small one. In chlorophyll A, you have a methane group, and in chlorophyll B, you have a CHO group. Very small functional group difference between chlorophyll A and chlorophyll B, but the consequence of that is that they absorb slightly different shades of green. A spectrophotometer is a device that measures a pigment's ability to absorb certain wavelengths of light. In other words, you can figure out what kind of light gets absorbed by a particular pigment. Here's the setup for a spectrophotometer. You take a white light source. The sun works, but usually in an experiment you'd be using a white light source, like a light bulb, and you pass it through what's called a refracting prism. This is more of a physics topic, but a refracting prism essentially breaks up light into its component parts. Remember that white light consists of all of the different colors of the visible spectrum. So when you pass it through a prism, what you see coming out is a rainbow. You then pass that rainbow through a very thin slit. Now the rainbow itself can't fit through the slit, but one particular wavelength of light will be able to pass through. That particular wavelength of light is then sent to a particular pigment. In this case, we're testing chlorophyll. And depending on how much of that light is reflected, we get either high transmittance, meaning lots is reflected, or very low transmittance, which means very little was reflected. Low transmittance means high absorption. So here, when you see green light passing through and getting reflected, you get a very high transmittance, which means the plant is not doing much photosynthesis if it gets only this wavelength of green light. Now down below in the second trial, we're filtering out the blue light. Now blue light will not be reflected by chlorophyll, it'll in fact be absorbed, and so you get very low transmittance. That means that a plant really could use blue light to do photosynthesis because it will absorb that blue light instead of transmitting or reflecting that light. Now, if you repeat this process for a lot of different colors, not just green and blue, but for a lot of colors, you can get what we would call an absorption spectrum. This is the kind of graph you'd want to be familiar with. An absorption spectrum shows you what colors of light a particular pigment absorbs versus what particular colors of light it reflects. So take a look here at chlorophyll A. You notice that chlorophyll A has very high absorption in the indigo and violet range. It sort of peaks there. And then it peaks again in the sort of red-orange area of the graph. Chlorophyll A, though, has very low absorption in the green area of the graph, the green wavelengths of light. What this tells us is what we already knew, is that chlorophyll A will not absorb very much green light. It'll reflect that green light, but it'll absorb colors in the indigo-violet range and colors in the red to orange range. You can trace this for other pigments. So here we see chlorophyll B, and this graph sort of explains why chlorophyll B would be an important accessory pigment. You notice that it peaks at slightly different places from chlorophyll A. Chlorophyll B peaks a little bit farther into the blue range and a little bit farther into the orange and yellow range than chlorophyll A does. That makes it a great accessory pigment because some of those wavelengths of light, which would otherwise not be usable by the plant, can be absorbed by chlorophyll B and can be used for photosynthesis. The same thing is true of the carotenoids. So the carotenoids pick up a lot of the wavelengths and absorb a lot of the wavelengths in the blue and indigo range that would otherwise be lost to chlorophyll A. So this is why carotenoids and chlorophyll B make for such great accessory pigments, because they absorb slightly different wavelengths of light and broaden the range of the visible spectrum that can actually be used by a plant. We'll come back to chlorophyll in just a moment, but first we're going to take a quick overview of the two stages of photosynthesis. As we said before, photosynthesis can be divided into the light reactions and the dark reactions. A quick note on the dark reactions. The dark reactions are not really a great name for this series of reactions because although they themselves do not require light as an energy source, they do require the products of the light reactions which require the input of light energy. So the dark reactions can maybe better be called the light-independent reactions, or summarized as the Calvin cycle, named after Melvin Calvin, who identified some of the chemical reactions that occur in this cycle. The purpose of the dark reactions, or the Calvin cycle, will be to build organic molecules. The thing is that requires a lot of energy, and that energy is obtained during the light reactions. So the light reactions of photosynthesis occur to trap light energy and convert it into a usable energy source that can be used to drive the Calvin cycle. So what does that energy look like? Well, initially it's light energy coming from the sun, but it's converted temporarily into NADPH. This is a reduced electron carrier that's carrying high energy electrons, and also ATP, which of course is that high energy molecule that can be used when a phosphate group is released. ATP releases a tremendous amount of energy. So NADPH and ATP are both created during the light reactions in order to drive the endergonic reactions that occur during the Calvin cycle. These are the reactions that build sugars. These are highly endergonic reactions, so they need the energy input from the light reactions. This next diagram is great because it shows you where all of these different things take place. So you can see that everything is happening inside the chloroplast. The light reactions occur in the thylakoids. Light strikes the thylakoids. That energy is then converted into ATP and NADPH. These products are then used to drive the Calvin cycle, the output of which is organic molecules like sugars, like glucose, for example. So let's get into the nitty-gritty of the light reactions. The question is, how exactly does a cell use light energy? We can't. Animals can't do this. So how is it that a plant cell with chloroplasts inside can use light energy from the sun? How do they capture it so effectively? Well, it has to do with chlorophyll. Here we have a depiction of a chlorophyll molecule. It's not very accurate in terms of the molecular structure of chlorophyll like we just saw, but for the sake of this diagram, here we have a chlorophyll molecule, and here it is struck with a photon, or a little bundle of light energy. That little bundle of light energy temporarily excites the electrons on chlorophyll. An excited electron is not a stable electron. It's actually very unstable. And so the natural tendency for an electron that's been struck with light is to go right back down to its ground state, to its non-excited state, to its stable state. And when the electron does that, it releases all of the energy that it just had. So it releases a photon of light and also a little bit of heat energy. Chlorophyll that's been isolated, so chlorophyll that is no longer in the thylakoid membranes in a chloroplast, will actually glow when it's hit with photons because all of the electrons that absorb that light energy, they go right back down to their ground state and they release these photons of light, which looks like fluorescence to us. So why do plants not glow? Well, plants need to be able to keep their electrons in their excited states. This is how they trap the light energy rather than just letting it go in the form of heat and fluorescence. So the chlorophyll that is inside plants, the chlorophyll that's actually used to do photosynthesis, is contained within photosystems. Photosystems allow that energy to be trapped. here's what a photosystem looks like. Well, actually, here's what a photosystem looks like. You can see here that we have the thylakoid membrane, and then we've got the photosystem. Now, this looks like a tangled mess, but you know some of the structures that we see here. We've got some alpha helices, especially in the region of the thylakoid membrane. We see some beta pleated sheets. And then embedded within all of these proteins, we see some green chlorophyll molecules scattered throughout. So this is a photosystem. It's a complex of lots of molecules, including proteins and chlorophyll, that's embedded within the thylakoid membrane. So, what is the photosystem? What is its purpose? Well, all of those chlorophyll molecules that are scattered throughout the photosystem, when they're struck with light energy, their electrons shoot up, so that their electrons get excited just like we would expect. The thing is that because this chlorophyll is inside a photosystem, that energy doesn't get wasted. The electrons don't just go right back down to their ground state and in the process release light and heat. They don't do that. Their electrons stay energized. So here you can see how it works. You've got a photon of light coming in. It strikes a chloroplast, or excuse me, it strikes a molecule of chlorophyll, and that chlorophyll has electrons that get excited. The electrons pass that energy to the next chlorophyll molecule, and they pass that energy to the next chlorophyll molecule. It's a little bit like a game of hot potato. So the energy keeps on getting passed on from chlorophyll molecule to chlorophyll molecule. It's just the energy so far. It's not actually any electrons that are getting passed from chlorophyll to chlorophyll. It's just energy. So the energy excites this chlorophyll first, then this chlorophyll, then that chlorophyll. So all of their electrons sort of get excited in sequence until finally the energy arrives at this special pair of chlorophyll molecules. They're called special because they can absorb energy and their electrons will actually leave. So their electrons will leave that pair, that special pair of chlorophyll molecules, and jump up to what we call the primary electron acceptor. That jump of these excited electrons from chlorophyll A to the primary electron acceptor is the first step of the light reactions. This is where energy has officially been captured from the sun by chlorophyll molecules and converted into a chemical form. Because now the primary electron acceptor has these excited electrons. Excited electrons are chemical energy. So we've got the transformation of light to chemical energy. And again, what makes these chlorophyll molecules here in the reaction center so special is that unlike all of these, they don't just transfer energy, they actually transfer their excited electrons. Now, if you think about it, that means that these chlorophyll A molecules will always sort of be losing electrons, and that's not sustainable. They can't always be losing electrons and still expect to continue to do that indefinitely. So we'll revisit that in just a moment. So hang on to that thought that this special pair of chlorophyll molecules is running out of electrons. But first, let's consider the two different kinds of photosystems we have. So there are two photosystems called Photosystems 2 and 1. This is a little confusing because light actually strikes photosystem number 2 sooner than it strikes photosystem number 1. So it's unfortunate that it's named that way. But we suspect that photosystem 2 probably evolved second, because photosystem 1 is used in many more forms of life, simpler forms of life like bacteria. So when you're studying the two photosystems and you're frustrated that photosystem 2 is used before photosystem 1, just remember that the numbers can also serve as a reminder to which one probably evolved first. We think photosystem 1 is older than photosystem 2. So how do these two photosystems work together? To answer that, I'm going to jump down to this diagram here. That does a nice job of summarizing the two photosystems. So notice that you've got photosystem 2 here, and the first step is light coming in and striking a chlorophyll molecule. Just as we saw before, the energy from that chlorophyll molecule, its excited electrons, gets passed to another chlorophyll molecule. and its excitement gets passed to another one, and its energy gets passed to another one, and the energy is passed and passed and passed until finally the energy reaches this special pair of chlorophyll molecules located in the reaction center. Now, this particular pair of chlorophyll molecules is called P680 because that's the wavelength of light that it absorbs the best. P680, 680 nanometers. The electrons that are excited now on this special pair of chlorophyll molecules get transferred to the primary electron acceptor molecule. So this pair of chlorophyll molecules, unlike all the others, not just transfers energy, but actually transfers its own electrons. And so now those excited electrons are with the primary electron acceptor. And I asked you to remember earlier that the special pair of chlorophyll molecules can't just keep on giving up its own electrons indefinitely, right? It needs to have a replenishment. Its electrons need to get replaced. And so this is actually where the water in photosynthesis gets used. Water is essentially a reduced form of oxygen. And so water, in this stage, right at the beginning of photosynthesis, gives up two of its electrons to become oxidized, and it forms oxygen as a waste product. It's funny to think that the oxygen from photosynthesizers, which we rely upon so heavily, to them is just a waste product. They just need the electrons from water to replace the lost electrons on this special pair of chlorophyll molecules in order to continue the process of the light reactions. But it produces this really valuable byproduct, this oxygen gas. Anyway, that's a bit of a side note. So these excited electrons passed to the primary electron acceptor. They've got so much energy. And what do we do with excited electrons? Well, you might remember from cell respiration that excited electrons can travel down an electron transport chain. The benefit of going down an electron transport chain is that the electrons have this opportunity to release a little bit of energy with every drop. A little energy is lost every time the electrons jump from one carrier to the next. So why is this exciting? Well, for one thing, this electron transport chain generates some ATP. We'll look at that more closely in a moment. But the electrons, as they go down the electron transport chain, finally reach their final electron acceptor. Now, in cell respiration, the final electron acceptor was oxygen. In electron flow between photosystems, the final electron acceptor is going to be another special pair of chlorophyll molecules. So this special pair of chlorophyll molecules in photosystem 1 is a little bit different from the special pair we saw in photosystem 2 because its electrons accept energy best at a wavelength of 700 nanometers. So we call them P700. By the time the electrons get there, they're pretty low in energy, but we are in a new photosystem, and so light can strike photosystem 1 just like it struck photosystem 2. The energy from light can bounce from chlorophyll to chlorophyll to chlorophyll until finally that energy reaches the P700. This special pair of chlorophyll molecules, having just gotten some new electrons from the electron transport chain, now gets excited and it shoots up its electrons to the primary electron acceptor of photosystem 1. From there, they go down a very short electron transport chain, and the final destination for these electrons is going to be NADP+. It'll reduce NADP+ to form NADPH. What's important is that this electron transport chain is shorter than the electron transport chain we saw between the two photosystems, and so these electrons are still carrying a good amount of energy. So, coming out of this process, we have some NADPH carrying high energy electrons. We've also generated some ATP. The overall consequence of this is that we have used light energy to generate high energy molecules, ATP and NADPH. Remember that these are the molecules that drive the dark reactions. These are going to provide the energy that's necessary in the Calvin cycle in order to drive the production of glucose and other organic sugars. This is the purpose of photosynthesis in the light reactions, to trap light energy and convert it into energy-rich chemicals, ATP and NADPH. Now, what we're going to look at next is just a couple of nitty-gritty details about this whole process. But don't lose sight of the big picture. We've got light energy coming in and ATP and NADPH coming out. Now, there's an alternative to this flow of electrons, which we call linear electron flow, because it comes in, the electrons come in with water, and they go out on NADPH. The electrons essentially travel in a line, linear electron flow. There's actually an alternative to this called cyclic electron flow that only uses one of the photosystems. It only uses photosystem number 1. It totally leaves out photosystem number 2. You'll see that's why it's grayed out here. What a great diagram. So photosystem 1 reuses its electrons. So instead of gaining electrons from water and then sending them off to NADPH, this cyclic electron flow reuses electrons in a cycle. That's why it's called cyclic. So in photosystem 1, light energy will still be captured by chlorophyll. But when the electrons are excited and sent to the primary acceptor, they're cycled to a short electron transport chain that generates from ATP, and then the electrons just go right back to photosystem 1. They go right back to that special pair of chlorophyll molecules, which gets excited by light energy, and then the electrons go back and repeat the process. So what's the outcome here? Well, we generate some ATP. We don't get any NADPH out of this process. And so we can't drive the Calvin cycle using just this process alone, but it does generate some extra ATP. So why would we do this? Well, sometimes you just need some ATP, and so this process will satisfy that demand for extra ATP when it's needed. But also, some organisms, like cyanobacteria, that do photosynthesis don't actually have photosystem 2. Remember, we think that photosystem 1 evolved sooner. So we suspect that these bacteria do cyclic electron flow and a form of photosynthesis that generates ATP, but not necessarily any organic compounds. So this is the process of cyclic electron flow. It's an alternative to the linear electron flow, which not only generates ATP, but also generates some NADPH, which can then be used in the Calvin cycle. Cyclic electron flow just gives you some ATP. Now, the other thing we're going to take a closer look at, if we jump back up to our summary diagram here, is step 4, the electron transport chain. Remember that energy-rich electrons that were on the primary acceptor of photosystem 2 travel down an electron transport chain and generate some ATP in the process. So let's take a closer look at how exactly that happens. This process is phosphorylation because we're building ATP by adding phosphate groups. And we call it photophosphorylation because that whole process is driven by light. So photophosphorylation. In other words, light is the energy source. Here's a very detailed close-up of a thylakoid. So we're not just inside a chloroplast now, we've zoomed in even farther. We're looking at a chloroplast, looking specifically at one particular thylakoid disc. And that's what you see here. This is the membrane of the thylakoid disc. It's a double phospholipid bilayer. And inside we have the thylakoid space. So, outside the thylakoid is the stroma. We know that already. Embedded within the thylakoid membrane, we have our 2 photosystems. We got photosystem 2 and photosystem 1. So now that we're oriented, let's take a look at what's happening in the diagram. These diagrams of the photosystems really just reflect what we already know. Light hits photosystem 2, electrons are excited until they eventually reach the special pair of P680 chlorophyll molecules, which give up their excited electrons to the primary electron acceptor. In order to make sure that P680 doesn't run out of its electrons, water is used to replace them. So water is oxidized when it gives up those electrons and is released as oxygen, a waste product. The excited electrons that are on the primary electron acceptor are carrying a lot of energy, so they travel down an electron transport chain until they end up very low energy in Photosystem 1. Photosystem 1 has chlorophyll, and so it can be excited by light just like Photosystem 2. That energy eventually makes its way to the P700 chlorophyll molecules, which has electrons that now get excited and jump up to another primary electron acceptor. Those excited electrons are eventually used to reduce NADP+ to NADPH. NADPH is now carrying those excited electrons, and they'll be used in the Calvin cycle. What's special about this diagram is it allows us to take a closer look at that electron transport chain that occurs in between Photosystem 2 and Photosystem 1. So this is where the excited electrons lose their energy and travel between this primary electron acceptor and the second pair of special chlorophyll molecules. So as the electrons travel down the electron transport chain, they lose a little bit of energy. Just like we saw in cellular respiration and oxidative phosphorylation, the energy from those traveling electrons as they jump from carrier to carrier is used to pump protons against their concentration gradient. You'll notice that the thylakoid space is colored darker and it has a high hydrogen ion concentration. The stroma outside the thylakoid space is colored lighter and it has a low hydrogen ion concentration. So if protons are to be pumped against their electrochemical gradient, they'll be pumped into the thylakoid space. Those protons now have a tremendous amount of potential energy. And when those protons rush through the ATP synthase molecule, this is a combination of a transport protein and an enzyme, they produce a proton motive force, which ATP synthase uses to make ATP. Just like what we saw in cellular respiration, the energy of moving protons is used to drive the phosphorylation of ADP to become ATP. And the ATP that's produced from that electron transport chain is ultimately sent to the Calvin cycle for the dark reactions. So the stroma has this low hydrogen ion concentration. That's why the hydrogen ions are quote-unquote happier there. They're lower in energy, they're more stable, and so in the stroma, they're happy to be there. They will rush through the ATP synthase out of the thylakoid space in order to get there. So that is the purpose of this electron transport chain between the two photosystems. At the end of it, we've generated some ATP for the Calvin cycle, and in linear electron flow, we also have generated some NADPH. That'll be brought to the Calvin cycle as well, and these energy-rich molecules will be used to drive the endergonic reactions that will build the sugars of photosynthesis.